thank you all for showing up so early to attend a Zoom meeting. My name is JSeegmiller. I'm a clinical chemist here at the university, for those that don't know me. This is my third grand rounds in 6 years, so hopefully I can get an award for that. But today I'm going to be talking about a project that I've been working on for actually a really long time, urine albumin, and how we're making the way to standardize this test. I have no disclosures for this talk. Just really briefly, I want to bring everybody up to speed on the ARDL lab. It's also known as the Advanced Research and Diagnostic Laboratory. It doesn't just do COVID testing, and really was primarily set up as a central biochemistry laboratory for NIH-funded studies. So things like GRADE, EDIC, Sprint, CDC, and Haynes. So ARDL was really created to sustain the same laboratory methods over a long period of time. And Dr. Mike Steffes and John Eckfold were really integral in seeing this and having this vision. And so what they did is they broke the clinical research away from the Fairview labs. And some of the reasoning for that, as you know, is the hospital administration can just pull a certain analyzer out and put a new one in for money purposes. But ARDL, for those that you don't know where ARDL is, so here this red dot is ARDL. Over here is the East Bank, and over here is West Bank, and you can, obviously this US Bank Stadium. So if you know on Washington Avenue where Bobby and Steve's Auto World is, it's right across from that. And there's also some of you may know about 1010 Washington Wine and Spirits as well too. So here's the lab. I'm not going to go over it too much, but it's a big lab, and there's just some pictures here in the accessioning area. We have a biorepository, and then down here is the mass spectrometry area as well. So the objectives today is you should be able to recall different measurement procedure approaches for urine albumin. You should also be able to recognize laboratory limitations and critical needs for urine albumin testing. And you should be able to identify necessary steps for measurement procedure standardization. So albuminuria basically occurs when serum albumin passes through the glomerulus at the rate which exceeds 30 milligrams of albumin per day. They also relate it to the amount of creatinine, so 30 milligrams per gram of creatinine per day. And the reason why they use creatinine is the 24-hour collections are difficult, and the volumes can be inaccurate. The patient doesn't collect all their urine over that time period. So the creatinine ratio they found is approximately equal to the urinary excreted albumin per day. And there's a link down here to the NIDDK that discusses this. But this is my favorite picture. When I think of the nephron, this is what I picture in my head here. So really, the current dogma between albuminuria and renal physiology is long thought that the glomerular basement carries this net negative charge. And this negative charge serves as a critical repulsive barrier to serum albumin to cross, since albumin is also negatively charged. But Miner pointed out that there's many studies that are coming up that suggest this charge argument is somewhat controversial. So a more recent view, what's happening with albuminuria and glomerular filtration, is that this paper from the New England Journal last year suggests that the glomerular basement membrane is a semi-permeable membrane and is controlled by buttress forces from podocytes. And what happens is these podocytes are injured, and the reduction of the filtration slit area makes a decline in glomerular filtration. So small solutes aren't able to clear, water can't clear, things of that nature. So your GFR goes down. Additionally, you have these buttress forces that are lost, and now you're able to increase your albumin permeability across the glomerular basement membrane. So this is the model that really gives a plausible theory to this mystery of like, okay, if it's just a screen, my salt, and I poke a hole in my screen, the small solutes are going to go through just as much as the albumin. But this kind of helps with that theory. Okay, I can have a low GFR and also be leaking albumin in my glomerulus too, as well. And so this is just that picture here. So over here is the healthy glomerular basement membrane. Here's the blood side. Here's the urine side. Here's your podocytes, and here's your slit diagram. So this is where the small solutes are going across in the slit diagram, and then the albumin small amounts of albumin will leak through daily in healthy people, but when you get disease, what happens? These podocytes are injured, and now you have less slit area here for small solutes to diffuse through or travel through, and now but the albumin can travel through the glomerular basement membrane more readily due to the decrease in the buttress force here. So okay, so now to some lab medicine standpoint. So the way that we, some common ways that we measure urine albumin are either dye binding assays or immunometric assays. So the dye binding assays are things such as BCP and BCG. So bromocrystal purple is a dye binding assay, and that's more specific. It provides a more specific reaction with human serum albumin. And actually, this is still performed right now over at St. Joe's from some of the Health East legacy Abbott Architect equipment. The difference between BCP and BCG is BCG can react with other acute phase proteins and potentially overestimates albumin concentrations. So most people will use the BCP as opposed to the BCG. There's the immunometric assays. A lot of times they're either nephelometric or turbidimetric. And the construct for these is there's an antibody that's specific to albumin, and typically they'll add something to those antibodies, like latex or some other reagent that causes it when it binds, it reduces solubility and creates this purulent, turbid mixture. And so when that happens, when that reaction happens, it scatters light. And so really the only difference between a nephelometric assay and a turbidimetric assay is the nephelometric assay usually detects the scattered light at perpendicular to the light source, and turbidimetry is usually 180 degrees to the light source. So the reason why I bring this up is our Siemens, what we're using now clinically, uses a nephelometric detection system, and what we're going to the Roche after we migrate uses a turbidimetric detection system. So what can we expect when we switch shortly here from the Siemens to the Roche? So overall, the urine albumin concentrations are likely going to decrease. And so below, I just put this package insert, and this is something, many years ago, I'd pull these package insert and look at the differences in reagents for urine albumin, and the vendors are kind of just all over the map. So I just highlighted, here's where we're at right now. The Siemens at level 1 would give us 86 mg per liter, and the Cobas, what we're going to at level 1 is going to give us 17 mg per liter, which is quite a big difference. And you go to level 2, you see similar things as well. The Cobas is at about 63, and the Siemens is more than double that at level 2. So you can see this big issue and sort of standardization and concentration effects between the different vendors for urine albumin when we have these set limits of 30 milligrams per day or 30 mg per gram creatinine. But one thing to keep in mind, there's a term called commutability, and so these controls are not commutable between manufacturers. So the way that what happens then is usually you want to look at something like a CAP survey, and so the CAP surveys are considered commutable matrices. So there's not just one sample that has an issue with a specific reagent system. When you pool the samples together or pool urines together, you dilute out those specific matrix interferences. So, and you produce a commutable matrix. But if you still, again, I'm just highlighting where we're going and where we are. So the Siemens, again, if you look at them between the Roche, they're still about 20% higher across the board between the different samples in this accuracy-based survey back in 2017. We published on this back then as well too. So when I really started to get involved with urine albumin, it was right around 2006 or so, but right before that, this paper came out by Wayne Comper, and he published this paper saying that immunoassays are only detecting immunoreactive albumin and are missing this what he called non-immunoreactive albumin. And he suggested the size exclusion HPLC is able to detect all forms of albumin, and the immunometric techniques are underestimating the total urine albumin. And so these are just some plots from his paper where it's showing two immunoassays here, and you have great correlation and pretty much identity scatter across the line of identity. So pretty good comparison study here. And then you compare one of these immunoassays to the size exclusion chromatography system, and you can see the line of identity is this dashed line here. And most of the comparisons are way below this dashed line. And again, this is on a log scale, so they're seeing stuff that's at 100 and on by size exclusion, and less than 10 by the turbidimetric method. So a big discrepancy here. So one thing to know about size exclusion chromatography, I just kind of pointed out the theory, is that small molecules basically go into these pores and get caught up. Okay, so they have to travel a longer distance through this pore, where the bigger pores, the bigger molecules can't enter this porous particle, and so they go around it, and they actually elute first. So the biggest things elute first, the next biggest things will elute later, and our smallest things will elute lastly. So there was another gentleman from the NIH that I worked with, Glenn Horton. We published a paper together, but this was from his group. And he found that the size exclusion method that Comper was looking at was unable to resolve several urinary proteins. And so what his lab did is he took albumin, transferrin, alpha-1 acid glycoprotein, alpha-1 proteinase inhibitor, and he injected it on the same method. And you can see there's a UV detector, so you really can't specify or get any more selectivity from that. So you can see the albumin overlaps with transferrin almost identically. You can see there's a little bit of shift here, but they're all coming off at the same spot. And if you mix albumin and transferrin, we're not even seeing like a little shoulder or something like that we might see in like a protein electrophoresis that we could not integrate. So really, this is one of the, it's one of several actually studies that suggest that there's not significant non-immunoreactive albumin in urine. And so the likely reason for these increased values observed is the size exclusion method is co-eluting other urinary proteins at the same time. So like everything else, once you want to get the right answer, everybody immediately goes to the mass spectrometry. And so Dr. Singh's lab down at Mayo Clinic came up with an LCMS method for urine albumin. But again, so that's important, LCMS, so he's not doing any tandem mass spectrometry here. Because albumin is a rather large molecule, and you're not able to fragment it in the collision cell. And so down here is a mass spectrometer rail, and here's your collision cell. But what they did do, so here's intact albumin here, and there's multiple charges, and you can see, and what they did is they cranked up the declustering potential or cone voltage here, and they were actually able to shear or produce this collisionally induced dissociation, or also known as in-source fragmentation, to produce these fragments here, which are shown down here. And the highest one, the most intense, they took this B24 ion, and then they produced an assay from that. But this B24 was not able to be fragmented in the collision cell, and that's why it's an LCMS and not a tandem mass spectrometry method. But when they made this method, they also manufactured this heavy albumin. So basically, they fed this yeast with minimal media, and in the minimal media, they had this heavy nitrogen, so N15. And so you do several iterations of this, and basically dilute out all the N14, which is the primary and naturally occurring isotope of nitrogen, and you enrich this albumin with these heavy nitrogens. So you can see here here's regular albumin, and here's the B24 ion, and here's the N15 labeled version of that. So there's 35 N15s in this B24, and you can see the mass shift here. So they have this internal standard there as well. So this is kind of the ideal case, and it was really making internal standards like this with proteins, with a fully intact protein, was really kind of ahead of the curve, and now most people are calling for that when you're doing total protein analysis. So one thing though with this LCMS method is, they asked me to look into it. The one thing about it is the sensitivity sort of tailors off at about 20 milligrams per liter. The other thing too, if you look at the line of identity here, most of the values are lower than the line of identity, and so there was a little bit of non-specificity. So proteinuric samples seem to have interferences that we couldn't really explain, and presumably this happened from other urinary proteins. So what we had to do then is we had to come up with something that's has much more specificity for albumin. So there's a guy by the name of John Barr that I think really pioneered this what's called now the bottom-up approach, and he was from the CDC. And in this approach, what you do is you digest a protein with an enzyme, and typically this is trypsin, but there are other enzymes, and then you examine specific peptides to the protein, and at that time they were coined proteotypic peptides. So you'd go and blast this peptide as a unique to your protein, and then you use that as a surrogate for quantitation of the intact protein here. So this is just a picture from his paper where he's using trypsin and he's pointing out different peptides that he was looking at. They didn't have a fully intact labeled system like Mayo did at that time, but what they would do is spike in heavy isotope labeled peptides, and those would serve as internal standards, which is easy, but it generally doesn't work very well. So that's when I came into play, and I was like, okay, well, if I'm going to have to use an enzyme to digest this thing to get the specificity that we need, I got to make every urine the same. Okay. The problem, the nice thing about John Barr and a lot of people that do things in serum and in blood is the blood is generally pretty much the same from person to person. You don't have these big pH swings, those sorts of things. The amount of protein is generally within a couple, or not even within, it's within the same order of magnitude. Whereas urine can go from 4 logs different in protein. Also, you can also have osmolality, really big changes in osmolality, big changes in the protein of interest, and then pH as well too. And so this is where one of the first experiments I did, where I just took 20 different urines and I looked at the pH, and I just looked at, well, what's the CV of the pH between person to person? He's 20, and you can see it's pretty high, it's 13%. But if you dilute it out with a buffer, in this case we use ammonium bicarbonate, you can see that you start to normalize really the pH of the urine. And then I think at about pH at a 15-fold dilution, you really normalize osmolality as well too. And so this really provided hope for me that we could normalize pH by dilution and be able to employ this enzymatic approach. So the way that we developed this method is we had the specimen and we had our N15 internal standard, and then we reduced it, and at that time we used DTT. What DTT does is basically you reduce the disulfide bonds, and then you unfold the protein so the enzyme can go and cut more efficiently. Another thing that you want to do as well too, or we end up doing too, is alkylating. So you alkylate after you reduce it, so these disulfide bonds don't form back, and so we have this nice unfolded protein that the enzyme is going to go and cleave. And then finally digest for 1 hour at 37 degrees, and then you end up with your peptides, and then you quench the reaction and inject on your mass spec system. So this is just kind of one of the peptides we looked at, and this is what you would see, an unlabeled versus an N15 labeled peptide. So here's our unlabeled peptide, and I put my sequence here into this calculator. And if you look, here is the mass that it should be. But when we look at this mass, what we actually see in the mass spec, we don't see this 1149 mass. We see this 575 mass. And the reason why we see a 575 mass is because this peptide is multiply charged. So that's why we see this 575. So we know it's multiply charged, and there's 2 protons on this peptide, giving it an M over Z, half of what this mass is. So if there was 1 proton on, this was what the mass would be. And remember, mass specs don't measure mass, they measure the mass to charge. And so that's why we see half of this. Okay, so we add 2 protons and divide by 2, and that's where we're gonna get this 575. The other neat thing, I think, is like you have your isotopic envelope. And the peak splitting between these two will tell you what charge state your peptide is in. So if I look between this mass, which is 575.3, and then this peak here is 575.8, that difference is 0.5, which gives me my charge state of 2. So charge state of +2, I have a 0.5 shift. If this charge state was 3+, this peak splitting here would be 0.33, etc. So, but the other thing to note is here is the elemental formula. So there's 12 nitrogens in this peptide. So I don't see a mass shift of 12 for my N15 label. I see a mass shift of 6 because it's divided by 2. So I have a 6 M to Z difference. The other neat thing that you'll see is there's some small N15 impurity. So if you have a 99% pure nitrogen source that you fed the yeast or insect cell or whatever it is, there's 1% that isn't pure. And so you see that in this peak right here. So our monoisotopic peaks that correspond to each other. And then here is a little bit of unlabeled from impurity here. So it's kind of a neat little thing. It's mathematical. It's actually quite striking when you start calculating these things and see these artifacts due to small impurities. So here are some of the results that we got in our study when we use this intact N15 internal standard versus the turbidimetric assay. And one thing about the turbidimetric assay is once you get below 20, 15, really a lot of these standard curves are not linear in those ranges, so you'll start to see things kind of fall off. So we knew that the linearity in this range falls off. You can get the same answer every single time because of the automation and stuff like that with those. But if you really do a good linearity study down in these lower levels, you generally fall off linearity with what the routine clinical analyzers are. And so that's where you see this difference here and sort of a lot of scatter once you get below 15 or so. But when you get above, and the terms that we used in this paper should no longer be used. So normal albinuric, we just call it albinuria, so less than 30. And then once you're albinuric, they call it microalbinuric. And they weren't really using these. These were used because of dipstick analyses, ranges and things like that. That's how they came up with these terms. So these really shouldn't be used anymore clinically. So again, this albinuric range above 30, and you can see we have really good correlation. There's a few outliers, and the reason why those outliers are there is because when they dilute this sample, it goes back down into this range here. So that's where we're talking about this non-linearity issue between for the turbidimetric method. But in general, it looks pretty good in this first study that we did. So the other thing too is we had 2 peptides at that time that we were able to get to work and validate in this work. And so in theory, the 2 peptides are surrogates for the total protein, and so they should equal one another. But this interesting thing that I was never able to explain this difference that we saw, and it always kind of bothered me. We had this great correlation here between these 2 different peptides, but it was off by about 18, 15%. And then once we get to higher concentrations, it comes back in line. But in this lower concentration, we'd see this this shift in slope here, and it always sort of bothered me. I was never able to explain it. We kind of again we didn't know what to expect. There wasn't a lot of papers out at this time that had any sort of information why this would happen. So we were able to push it through in publication, but it always was in the back of my mind why this difference existed. So to kind of summarize the Mayo Clinic method that we developed, moving to this bottom-up approach really increased our sensitivity and our specificity for urine albumin. We could get down, instead of 20 mg per liter, we could get down to 3 mg per liter, which is plenty low enough for routine clinical samples. And we really didn't have any of these mass interferences in highly proteinuric samples. And one of the criticism is we only had 2 peptides that we were monitoring, and people wanted us to have, everybody thinks that you can have 20 different peptides and things like that. But when you start diving into it, there's a lot of complications, post-translational modifications, other sorts of things that happen in these proteins that, may be different from person to person. So We finished this, and the reality of the situation is this method was too time consuming for routine analysis. So the turbidimetric assays on the chemistry analyzers are interfaced. You have to do a digestion, alkylation, and reduction. Those things take 5-6 hours, and then you got to manually put the stuff into a spreadsheet and then upload it into your LIMS system. So it just wasn't feasible for a high throughput clinical lab or reference lab like Mayo Clinic has. So that was when they're like, okay, well, maybe it's not this routine lab, but we can do a reference measurement procedure. And so that was attractive since it didn't have antibodies. So always the vendors with the different methods will be like, well, my antibody works better versus your antibody. So they have this debate that no one can ever solve. But here's a system that doesn't have any antibodies. And so whenever you say reference measurement procedure, it triggers this person called Greg Miller to get involved. So Greg Miller started getting involved, and there's this other guy at National Institute of Standards and Technology by the name of David Blanc. And so there was lots of meetings and lots of things happening. It was really hard to get the wheels turning, but it was shortly after the publication that I left and headed into industry. But then I came back to school here at the University of Minnesota. I enrolled in the fellowship program in 2014. And so one of my mentors and good friends, Dr. Eckfeld, goes to the CAP meeting, and he comes back and he's like, Jesse, you need to restart this urine albumin work. And I was like, well, John, there's a problem. We got to get this N15 labeled albumin, and it's expensive and those sorts of things. And so we tried wing peptides and all sorts of cheaper approaches, and it just didn't work. And there's a reason for that. We tried to work with Mayo Clinic to have them make it again, and they were unsuccessful. I also tried to work with people here on campus, and I was just really unsuccessful and getting anybody to help us on this. I went to the NMR facility. They make a lot of N15 proteins. And then finally, I found a commercial vendor who does make this. And we still had this problem with no funding, but we were able to get a small amount of funding to eventually validate the method here in Minnesota. So more recently, we came upon an NHANES contract where they don't want to use the turbidimetric methods because they know that there's sensitivity issues below, say, 20 mg per liter. So previously, and for forever, they've been having these run at the University of Minnesota, but not at ARTL. It was in Dr. Blanche Shaver's lab. And so this was a homebrew assay that Blanche Shaver's lab brought up. And it's a fluorescent immunoassay, and it was performed manually with 2 technologists. And recently, Dr. Chavers retired, and the equipment is really not transferable. It's way out of date. Had that nice almond color on it. You can't get parts for things like that as well. So we really couldn't bring this into ARTL. So that's at that point, NHANES approaches us, and they want to use the LCMS MS method for specimens. So now I finally have a secure funding source to pursue, standardization along with routine analysis. So the difference in the measurement procedure as opposed to the Mayo method is we added 4 more peptides down here. But what we had to do first is we had to validate it, and we did this single measurement and just see where the imprecision lines up for these different peptides. And when we look at these 6 different peptides, we're also following the analogs for the 15N labeled peptides here. One different thing that I changed with the method is I ended up using TSEP as a reducing agent rather than DTT, and that was from some of the insulin work we did and how much better this was a reducing agent than DTT. So after I validated this method, I found that it performed at about 4 to 8% CV, which for mass spec is really good. And that's throughout the concentration range. So I was feeling pretty good about that, but we went back to the NIDDK lab working group, and they're like, no, it has to be better than the immunoassay system. I'm like, well, that's just not going to happen. But they came upon a metric. We have to get better than 5% CV for a reference measurement procedure. So Yeah, that's a problem. With mass spectrometry, if you have an auto sampler, is going to give you 2% CV just from injections. But we discussed and we came upon a consensus that we would next develop the same measurement procedure with replication of 4. So the square root of 4 is 2, and that'll improve our imprecision by a factor of 2 to potentially achieve this less than 5 metric that we're looking for. So here's a visual overview of where this is just the albumin sequence, and here's where the different peptides are located at that we chose. So this is without the signal peptide in front. And one thing to note too, and I always try to be open and honest when we do protein quantitation, is there could be other isoforms of proteins, and albumin does have different isoforms documented. And then there could be some sort of post-translational modification or something else that happens that inhibits enzymatic digestion that could cause issues. So that's one of the limitations for this type of approach as well, even though it's antibody void of antibodies. So here's our method. So even though we're looking at 6 things, we look at 24 different MRM. So here's kind of a neat example of a peptide. So you look at a peptide. So here's what we're looking at in our first quadruple of our mass spec. And then what's interesting, and we for this peptide, sometimes what you can do is you can actually look at a higher mass. So you go from a low mass to a high mass. And the reason why you're doing that is you go from a doubly charged species to a singly charged species. So we're looking at the Y6 ion of this peptide here. So that would be this guy. And so the nice thing about peptides is you can predict really nicely and with your empirical experiments and see which peptide and which transition you're looking at. So in this case, we're looking at the Y6 ion. And then what I like to do too, I always try to do this, is I take my internal standard, I look at my internal standard right after my analyte of interest, whether it's small molecule or peptide. So I do the Y6 IS. And so we see that same thing. We see a mass shift of 5 and a mass shift of 8 in the product ion. So we're looking at the other thing that I do too, or you try to do, if you are able to, is you can get 2 different ions, and you can do ion ratio. So if there's interferences and things like that in your method, you use this ion ratio. The ion ratio should be the same no matter what concentration it is, but if an interference happens, it'll make that ion ratio come unbalanced. And so you can be like, hey, maybe there's an interference in this specimen. So that's why we try to get 2 per specimen or analyte. But you can see so instead of having just 6 things, we have 24 things. So every injection has 24 points that you need to look at. So a lot of data. And so here's a chromatographic example. Again, I'm not doing any I'm not going to read any words from LabCorp at pushing things through really fast, and I'm not dragging things out for an hour and a half either. But we have nice separation, equilibrating the column well. But you can see here's what the chromatograms look like. And while it looks like this, single chromatogram, each one of these things is actually independent. So we can extract those. So if you have an extracted ionic chromatogram, even though when we look at these things, they're independent, we pull it out and it'll be by itself. So I just pulled this out. So there's an internal standard and then the analyte of interest of this peptide here. So each one of these we look at in quantitation. So now calibration curve. So a lot of times in the routine lab, when we ask them to calibrate, they calibrate once a week or something like that. We're calibrating every day, and we're doing 12 calibration curves for every batch. So this is kind of what that looks like. Again, a lot of data in these methods here. So here's an example of the quality of data that we're getting. So here's our peptide of interest, and over here's our accuracy. So between most of these between 95 and 100% accuracy, but there is one that goes down to 7% accuracy. But this is a nice linear calibration curve. I wish all tests look like this, all mass spec tests look like this, but they don't. So we have a good system, but we have to do 4 more injections now as well. And so the other thing that happened right before the pandemic is NIST released this standard reference material, or SRM 2925. And this could be used to value assign calibrators. So given this, the problem with this reference material is there's only a 0.5 ml of it. I wish that the concentration would have been really high, because then I could make my calibrators directly for it. So now there's a big problem. I have to make 2 years worth of calibrators or something like that I can store in a -80 freezer, but I can't make it with this NIST thing. So what I have to do is I have to value assign things that I make using this NIST thing. So there's another process that needs to take place. So here's our first go round. We take this NIST certified reference material and we make calibrators at these levels here. And then we analyze these calibrators in quadruplicate. And then I prepared, in-house calibrators from the Sigma Aldrich serum albumin that I purchased, that I was using, 12 years ago when I did the work at Mayo. And they characterize that as a, it's serum. It's about 99% pure. They suggest. And so we make some target concentrations of from 3 up to 200. So here's where we looked at the NIST calibrators, and we perform calibration across the different peptides. We have really good concordance with the different peptides and have reasonable accuracy. So we don't really see any peptide stick out over the other. They're all within sort of the error that we expect. So now when I look to the value assignment results, though, when we're looking at here's what the Sigma calibrator should be, and then we reference that against the NIST calibration curve, and then they come back. We start to see these, about a 10–15% difference among a lot of the different calibrators. But the one thing that really struck me as odd is there was this peptide that's about 15% lower than these other ones as well too. So even if they're off, I actually expected it to be off compared to the NIST reference material. But what I didn't expect is them to be off from each other. They should be the same. So this was suggesting that there's something else going on here. And the interesting thing is that when I remember back to that first paper that we published, we saw the same sort of thing with the same peptide with these two peptides. There's about a 15% difference here. So this suggests that Sigma Aldrich stuff may contain different levels of fragmented human serum albumin. So that may partially explain why we saw what we did back then, giving the albumin calibration material that we used between these two different peptides. So, okay, so now we're like, oh, well, if we have fragments, that's no good. We have to find a different source of albumin. So I found this company called Albumin Biosciences, and I think that they perform infusions in humans with their products. So they have really good manufacturing in place. But it's a recombinant source. And so we once again made our NIST gravimetric calibrators, and then we value assigned these new sources. And now this time we didn't see those issues. And what we saw, here's what we expected when we made our new calibration source, and this is the difference that we observed from what we expected. So it's really close to what we expected, and there was no difference now between peptide to peptide. So this new stuff, the point is the Sigma stuff in your calibration material could be very important, where the Sigma material had these albumin fragments and the NIST pure material and the Albumin Bioscience material did not have these albumin fragments. So now here's my new calibration curve with 4 separate injections at each calibrator with all 6 peptides examined. So basically what we do is those will sum up an average at each point 4 times, and it'll produce the average of those calibration results. And so this is what this is just one peptide, 1 of 6. This is the type of data that we see every single day when we run this method. So again, we have really good accuracy and precision, but we're going to average these 4 points and produce a single value at that point from 3 to 200. Here's a specimen comparison. Now, this wasn't running quadruplicate since I don't have IRB approval to do that yet, but this was for single measurements. And again, we see this sample comparison, and it's actually pretty good, but there's this outlier here, kind of similar to what we saw in my first publication. But this is, flash forward 13 years or whatever. So if we take that flyer out, you can see, look at the correlation here. It's just phenomenal. We expect there to be some slope differences because the calibration is likely different between the Roche and stuff like that. But for urine albumin to have a correlation, a sample comparison plot that looks like this is just, really phenomenal. It's a really tribute to the ARTO lab here and what they've been able to accomplish. I've been a part of Biocon, and you should see the correlations that we see between methods in that study from what I've seen. But again, this was only done at one time. If anything, this correlation will only improve if we run it for each sample 4 times. So this gives us hope, though, too. I mean, that's the other thing. So the goal here is to standardize these methods. As you saw, we can have really big differences between hospital systems depending on what reagent system you select. So this gives us hope that, hey, they're probably detecting the right thing, but if we can get the calibration systems standardized, likely we have a good pathway to fixing this globally. So for my future work, I still need a final comparison study for the LC-MS candidate they call this, instead of assays, they call them candidate reference measurement procedure. So if you see me talking about that, that's the jargon that you have to use. So we have to do a replication of this, do the 4 replications, and that's how it's going to reduce our CV by a square root of 4 of our replications, which will reduce it by a factor of 2. So it's going to get us below that required need for a reference measurement procedure. So I'm waiting on the IRB, but after I do that, we'll publish it. We'll compare the Siemens Roche to the mass spec method. And then I didn't know this. I didn't know I had to publish this. I was actually waiting to publish with the consortium, but they want you to publish independently. And then after you publish it, you can list the method with what's called the JCETLM, or the Joint Commission for Traceability and Laboratory Medicine, as a reference method. And then after this happens, then we can perform sample comparisons amongst other candidate reference measurement procedures. So other laboratories that have what they call a reference measurement procedure, and this will happen as they come online. So the pandemic, most people have not even started working on this, or if they have, they just started, like Mayo and NIST. And there's also a Japanese laboratory that has a method also. The thing that I guess is very important is you have to have 2 laboratories active for these reference measurement procedures, so customers have a choice. It's kind of a bad business model for, like, if you're the laboratory, but it's so that Beckman Coulter can go to Mayo Clinic, or Beckman Coulter can go to Minnesota and do these sorts of things, or Roche, or vice versa. So one thing about this comparison study is a lot of these other labs want to just perform like 10 sample comparisons. And I kind of was like, well, I don't think that's really feasible, because to do statistics, you almost need 10 samples anyway to do the minimum amount of statistics. So I spoke up and I made them do some power calculations. So we're going to determine the number of specimens for this round robin comparison once we get the power calculations back and all these other labs come online. So that'll be coming hopefully this summer sometime. So kind of for some concluding remarks, there's several different ways that we can measure urine albumin, and they each have their own unique limitations. The LC-MS candidate reference measurement procedure is going to proceed, and the validation is complete except for our sample comparison study. And then I'm going to publish this after I obtain IRB approval, and get the final sample comparison study between the Roche Siemens and the mass spec method. Value assignment of in-house made calibrators was achieved using this standard certified reference material from NIST. As you saw, the Sigma Aldrich pure albumin was not pure; it had fragments, and the data that I showed today suggests there's likely fragments in this starting material. So you have to be cognizant of these things when you go out and perform these types of experiments. And a lot of times you don't know it until the 11th hour, like myself. So we were able to fortunately find another stock that did have intact albumin the whole way. So, and then finally, once we get the specimen comparisons among the other candidate reference measurement procedures, that'll be sort of our conclusion. And we're on the road and list with JCTLM. So I'd like to acknowledge several people. So Mohammed really did a lot of the initial albumin work. Kang did a lot of the replicate analysis. So Mohammed recently left the laboratory. Kang is still here. He did a lot of the replication work, 4 injections per every sample, every calibrator or control. Dave Schmidt, who also departed this last fall in my lab, he was really instrumental in value assignment stuff as well. And then, John Eckfeld has really always been there for me in my career. So I'm very thankful to have him as a mentor and forcing me to look at albumin again. So I really appreciate him. He's always, like I said, he's always been there for me and put me down the right path. And Greg Miller is really instrumental too, because he's done this work with creatinine and things of that nature. So he's really overseeing what needs to be done administratively, and we have all our bases covered when we go into this final standardization. So, and finally, since we're all talking about how we don't see each other, so I figured I'd throw some pictures of my family. Here's my daughter. She's 5'8.5" now. We did a lot of lacrosse last summer. I think we had 9 different tournaments we attended. Sydney's also, she just left today. Actually, Courtney and Sydney just left today for Florida. They're going to cheerleading nationals down in Orlando. Here we are. We took a flight, helicopter flight in Hawaii on our vacation. While we're in black, we're not goth or anything like that. They recommend black so you don't have reflection off the windows when you see the stuff. And here down here is Trent's team got 3rd in state this year in lacrosse. There was a particular parent coach in the back here that was instrumental in that effort. So with that, I take any questions, and thank you all for listening.
Jesse, this is John Crossan. Fascinating talk. Thank you so much. We just are finishing up the medical school course in HHD4 in nephrology. And so I've been working with the nephrologists, and I have several questions to ask you. Sure. The nephrologists are absolutely committed to the upper limit of quotes normal albumin being 30 mgs per day, and the difference between 30 mgs and 300 mgs represents to them microalbuminuria. And I hate to tell you this, but they still use that term. My question is, number one, when you switch to Roche, is this going to change their concepts of these 2 numbers at all? Because they're really committed to those numbers.
Exactly. That's a great point, John. And that's the problem, right? Because when we do switch to Roche, they're committed to the numbers, so that they're committed to those numbers, but the real actual numbers are going to change, right?
Yeah.
From method to method. So
right.
That's the problem right now. If we go back and we look at those values from method to method, that's the real problem that they don't really know. And I think what will happen eventually, once we get these things standardized and stuff like that, they're going to have to reevaluate, maybe even have to reevaluate those hard numbers that have been ingrained for 25 years or whatever.
Yeah.
Right.
Yeah.
So that's a long road, and that's going to be a lot of education. I think there's going to be some studies in the ARDO lab that actually we reevaluate, is 30 the cutoff, or is there another cutoff after we get the right, concentration of albumin?
So the other question basically has to do with quotes normal amounts of albumin, or albumin levels, urinary albumin levels in quotes normals. And do you know where this 30 mgs per liter or 30 mgs per day came from? And secondly, with your new ARDL technology, is this going to allow us to learn a little bit more about the filtration barrier and how it manages albumin? Because there's been a lot of knowledge, as you indicated, generated in the last 10 years or so about the filtration barrier, but there's still a lot of questions about it.
Yeah, in terms of like the filtration barrier, I don't know, I don't know if we're going to be able to see, make those kind of delineations, because we're just looking at albumin. And so if we were looking at other things, maybe sloughing off, other proteins maybe sloughing off, that might be a clue, podocyte destruction or something like that. But we're just not doing that in this method. It's not that it can't be done, but I know there are groups looking at those sorts of things, specifically in like preeclampsia and things like that. So they're trying to look at, other proteins that would say, okay, well, there was damage occurring, and so they look at those sorts of things. But going back to your other question, where this 30 came from, I'm not sure. It's been around. I've also thought that myself, but it probably came out of the Ardo lab from some study, to be honest. But it was a long time ago that was established. And like you said, I think the problem, once you get above that 30 mg per day, that's pathogenic. So microalbumin, I think that's the problem, is some people think, well, it's microalbumin, it's not really bad. And I think that's what the people want to get away from, because if you're greater than 30, that's a problem. You have kidney disease.
Yep, definitely. Yeah. So another question. Recently, nephrologists have come up with a theory that edema in nephrotic syndrome in minimal change disease actually comes because there are some other proteins filtered along with albumin, and in particular plasminogen, which is converted to plasmin in the urine, and as the urine is being formed, and increases a sodium pump in the kidney. Do you measure plasminogen at all in the urine, because it's about the same size as albumin?
Yeah, no, so I'll go back to the method here. So basically, when we create these methods, you only get what you're looking for.
Yeah. Okay.
So it's very specific. So in my method, I'd have to put in, these other molecules and things like that I want to look for, other proteins that we look for. And so what we do here is basically, I only look for this, mass, and I look for this mass, and it's specific to this peptide here. So any other peptides, I'm blinded to. It's not like I'm have it open and I'm scanning all these different masses and stuff like that. So this is a targeted method as opposed to like a survey method or an untargeted method where I'm looking for unknowns every single time. So I'm very targeted and specific in what I want to look at. I have blinders on. I'm only looking for albumin, and I'm looking for these peptides here. So, but in order to do that stuff, we'd have to, either open it up to an untargeted sort of experiment, or add those targets into our current experiment here.
Okay.
Great questions.
This is Leela. Does the urinary albumin have bound fatty acids also, or and would that affect the methodology for analysis?
I mean, so you can have all sorts of different, albumin is a molecule that primarily binds lots of drugs and things like that, right? It's one of those compounds that does that sort of thing. I think once we perform a lot of these, and I think that's probably why some of these peptides, not all peptides work, right? So there's likely some post-translational modifications, those sorts of things. So what you want to do when you select, at least in this approach that we've taken, we selected peptides where there's likely minimal issues with these peptides. That's not to say that there couldn't be. I never say never anymore. But if there is something that we know that's highly has lots of post-translational modifications, we kind of move away from that. Or if there's differential modification of that part of the protein, we would move away from that and try to grab a peptide or a section of the protein where there isn't issues that are variable from patient to patient.
Thank you.
Jess, I had a question about the involvement of the IRB. It would seem surprising to me, although it probably shouldn't be, that the IRB gets involved with laboratory quantitation and normalization.
Mm-hmm.
Is this a difficult area for them to evaluate?
No, usually, so it should be minimal. It's a minimal risk study, right? We're just going to take samples that have previously been collected. But one of the things that's come up is in the last few years is if you're going to publish anything, you should get IRB approval. And so it's just one of those things that we just go through. And like I said, if I knew I had to publish this method to have it listed, I would have done that IRB approval. But I just found out like a week and a half ago that I need to publish this on my own and in order to do that, I got to get the IRB approval. So we're working on that right now. I'm assuming it should happen in a week or two. We'll get that approval and then we'll finish the study up. But yeah, for like a routine, if this was going in my clinic, right? If this was going in our routine lab, this is coming over at Artle, which is not the clinical lab. But if this is in my clinical lab, I won't get any IRB approval because it's the mode of operation, unless I want to publish that data.
But you also have to get CLIA approval, do you not?
Well, we're CLIA certified, so I follow the CLIA guidelines in terms of the validation requirements.
Okay.
And then we get inspected by,
Thank you.
Yep.
Anyone else? That was a great talk. Thank you so much. When I was thinking about John's question, these clinical decision cutoff points are, as you know, frequently selected without any consideration of what's feasible in clinical labs. And in the case of troponin, as you know, they went with 99th percentile of what's considered to be the reference interval. Anything above that is considered abnormal. Have you come across anything like that in terms of in a assay that's generally across commercial assays is poorly harmonized, like urine albumin? Any discussion about that approach rather than using a hard?
Yeah. I mean, I think when you look at the different vendors, we have this for urine albumin. Let's say we have this ingrained 30 level by nephrology. But if you actually look in the package inserts, I think in the Siemens and likely the Roche, I think those cutoff levels are a little bit different in the reference intervals. When they've done their clinical studies and determined their reference intervals, I think they're a little different than that in each one. So the manufacturers are doing some stuff, but again, there's not a lot of reason though. I mean, the other problem is there's not a lot of reason for them to work with each other, right? Because right now they can say, well, mine's better than yours. So once we have this reference measurement procedure, the thought is we're going to focus them in and then come up with these better clinical decision points.
Yeah, that's what happened with the serum creatinine.
Yeah, exactly.
Standardized assay. Then some manufacturers, it was clear that their assays were running too high in most cases.
Yeah. And Siemens, unfortunately, the Siemens is I think one of the higher ones for urine albumin out there, which is funny too because they have different versions of it, right? And so some are high and some are closer to others as well. So it'll be a big project.
Yeah, there's John Eckfeld. Yeah, I know Biocon study, which is a consortium of multiple studies that Jesse's involved with, is actually thinking that maybe a lower value will have prediction of outcomes in diabetes and other disorders. So, but the problem has always been that the clinical available methods were really not very good at the lower concentrations below 30. I mean, and they've not only varied a lot, their precision from sample to sample weren't very good. So I think they're hoping that this type of standardization and a more accurate measurement will lead to some clinical outcome prediction information.
And that's where I think IRB would like to be involved.
Well, all of these studies have IRB approval for collecting the samples like that. I mean, the diabetes control and complication trial, the Crick study, and they have stored samples, tens of thousands of stored samples that already have information on outcomes. So the idea is to go back and look at the information.
Wonderful biorepository.
Right.
Yeah, John, are you suggesting that maybe when we begin to do these better assays and more reliable assays, that number 30 mgs per day will be lowered, particularly in diabetics?
Yeah, I think that's the supposition, but they don't have the data to get prediction. Right. And it'll probably be graded. The higher the value, the more likely there'll be renal deterioration. But there may be information at lower values that they don't have information yet because they don't have the data.
Right. Gotcha.
Okay, I think we are going to close up today. It is 9 a.m. We will see you next Wednesday, and next Wednesday at 9, we have our QA/QC meeting with HemePath. Thank you very much, everybody. Have a good one.